room connecting the other.
இருங்க தம்பி வரும்.
ஓகே, தேங்க்யூ. ஸ்டீம் கரெக்டாக இருக்கு சார். லைவ் சாட்டில் போட வந்துருந்துச்சு, அதில் போட்டுடலாம். நான் போடுவேன்டா, நான் தான்டா போட்டுறேன்டா. இப்போ நீ யூடியூப்ல பாரு, ஜிமீட்டில் வந்து 100 தான், அதனால நாங்கள் யூடியூப்ல நடத்துறோம். நீ யூடியூப்லேயே பாரு.
நீங்க மட்டும் தானே சார், வேற யாரும் இல்லை, வேற யாருமே இல்லை, நீங்க தான் பேசுறீங்க, டைரக்டா நான் பேசுறேன். ஒரு சின்ன இது மட்டும், ஏஎம்ஆர்னா என்ன அப்படி சொல்லிட்டு ஆன்டி மைக்னே சொல்ல இனிஷியேட் பண்ணும்போது வெல்டெக் ஹைடெக் தேங்க் பண்ணிட்டு பிரின்சிபலை தேங்க் பண்ணிட்டு ஆஸ் பர் நார்ம்ஸ் அதை நான் பண்ணிட்டேன். இது பவித்ரன் பண்ணுவான் ஸ்டூடெண்ட்.
குட் ஆஃப்டர்நூன் சொல்லிட்டு
பண்ணிட்டு உங்களை நான் அவன் கூப்பிடுவான் உள்ளார, அதுக்கப்புறமா நான். ஓகே,
இதில் வந்து
குட் ஆஃப்டர்நூன்
இந்த ஆர்கனைஸ் கான்ஃபரன்ஸ் படி சொல்லிட்டு இந்த பிரின்சிபல் போட்டிருக்கேன். பிரின்சிபல் இப்போ வரல,
பிரின்சிபல் டேட்டா யுவராஜ் சார். சரி, நான் என்ன சொல்லிட்டேன் இது மட்டும் சொல்லு. Good afternoon, one and all. We are happy to.
With immense pleasure, I welcome you all to a 3-day popular lecture series organized by Department of Biotechnology, sponsored by Ministry of Health and Services, Government of India. Next, I welcome our respected Dean, Dr. D. Yuvraj, and Head of the Department, Head of the Department, Dr. K. Nanisekharan. I will also, இது உங்களை பற்றி இது. Good afternoon to one and all present here.
Happy afternoon, everybody. On behalf of the Department of Biotechnology, Veltech High Tech, Dr. Rangarajan, Dr. Saguntala Engineering College, I welcome you all for this 3-day popular lecture series, a sponsored event on antimicrobial resistant research. And the next 3 days, you are going to hear lectures on this particular topic, which is trending now in current research. So I definitely assure you that you'll be gaining insights into this particular topic on antimicrobial resistance, which is of greater importance right now after the incidence of COVID pandemic. So definitely hope you all agree to me in listening to these lectures. And today I think Dr. Sai Ramesh will initiate this particular.
So good afternoon to one and all. I hope I'm audible to you all.
Yeah, sure.
Yeah, thank you. So it's good being with all the young minds. So my presentation, without much further delay, it will be on antimicrobial resistance research, which is the need of the hour. So the whole PLS is being structured in such a way that we concentrate maximum on the antimicrobial resistance research, which is the dominant for the next few years in both science and pharmacology oriented domains.
Okay.
So myself, Dr. A. Sai Ramesh, Associate Professor of Department of Biotechnology, Veltech High Tech, and this particular PLS has been funded by DBT, and I thank both the DBT as well as the Veltech High Tech management for providing me this opportunity. So over to the research part. So I just wanted the Google Meet people to tell me what this particular arena is. Can anyone figure it out? What it is? Any idea? Can anyone tell?
WHO.
WHO.
Facts. Yes. 72nd World Health Assembly, and most of us would have seen this particular person, and he is the Director General. He has given the uh, initial flag mark. Red flag has been raised, where he says that the by the end of 2050, the number of deaths, the mortality caused due to antimicrobial resistance pattern would cross the uh, mortality rate, what could be the mortality rate for AMR in 2050, which is nearly 10 minutes. And what my research is totally on it, and how I deal the antimicrobial resistance research in my laboratory. And finally, I conclude today's session and leave the session open for the other three members on a similar arena who will also speak on antimicrobial resistance. So basic introduction, that is why the resistance research has one such examples is your bacterial pneumonia, urinary tract infection, all these things where your antibiotics are being involved. People say even cold, they try to give antibiotics, but antibiotics never reduce your cold. Uh, Cold, it takes eventually a time to. Topic is your antimicrobes, a broader term, which establish no basic selective inhibitions. A few examples for it is these two terms are used parallelly. One is antibiotic microorganism, which survives and grow continuously even after exposing it to certain antimicrobials. This may be a pattern where one particular antimicrobials would have been shown to be resistant in the past, where the antimicroorganisms would have developed a resistance pattern in present day or may develop a resistant pattern in future. This particular phenomena is called as antimicrobial resistance. So the question is it going to be a concern? Uh, Many of you would have guessed who the person is. He is not other than our Nobel laureate, Alexander Fleming. He predicted this in 1945 itself, where he said that if these antibiotics in 1945 by the inventor itself, the Nobel laureate Sir Alexander Fleming.
In 1928, antibiotics have saved millions of lives around the world, and the average life expectancy has increased considerably. But the wealth of these miracle drugs is not only a success story. The bacteria have fought back, and today we see an alarming trend. The number of multi-drug resistant bacteria has grown dramatically over the last decades, resulting in thousands of deaths due to infections that cannot be cured. What happened? The massive use of antibiotics has stimulated bacteria to develop resistance mechanisms in order to survive. For many years, lack of access to antibiotics actually causes more deaths than resistance. Multi-drug resistant bacteria spread within hospitals as a result of poor infection control, and also between people in the community. Bacteria know no boundaries, and extensive international travel and trade help resistance spread from continent to continent. Antibiotic resistance does not only affect humans, but also the health of animals and the environment. It is all connected. Be at higher risk for untreatable infections. Resistance is costly, both for the individual and for society. By 2050, the number of deaths due to antibiotic resistance could reach 10 million per year, and thus exceed the number of people who annually die from cancer today. Antibiotic resistance has been considered as dangerous and deadly as terrorism and global warming.
Explained here has already been conveyed through you in one short video of 2 minutes, where this picture clearly depicts the actual conversation that happened for the past 2 minutes. Since 1987, we are lacking, or we are out of antibiotic discovery. So now there is a space or a research gap which needs to be filled on. and the result of not having new antibiotics has raised the concern of certain operative procedures and cancer therapies where we are looking for a potential antibiotics so that the treatment becomes safer. So this leads to the second topic, how superpower became superbug. So this will clearly help you to understand how these antibiotics now became or now raises the concern where we are looking into a factor called as antimicrobial resistance. So antimicrobials usually have certain level of mode of actions. Usually these antimicrobials try to kill the other microorganisms in certain formats, in certain way of attacking the other microorganism. One is concentrating on the cell wall. List of antibiotics are given there or displayed onto your screen. The next is human being and also in other organisms. Certain antimicrobials are capable of killing viruses, which has also been shown to you over the screen. And we could see that the sulfonamides extend its arms, prescribe to the candidates even without proper screening, which have been done prior to the prescription of these particular drugs. Whenever a doctor is being addressed, when one particular antimicrobial or when one particular antibiotic doesn't work out for the individual, it is mandatory to check the antimicrobial screening before they progress for the um, for the prescription of second level of antibiotics. Usually we all would have heard that the first line of antibiotics has to be given, and before administrating the second level or prescribing the second level of antibiotics, it is always advisable for the doctors to check the antimicrobials and then prescribe the specific antibiotics to the patients. But not all doctors practice this particular pattern. Because of this, what happens is a particular candidate initially takes one set of antibiotics who doesn't even fulfill the complete dosage pattern what the doctor would have said, and if his or he tries to move to the another doctor and takes a separate or a different kind of antibiotics, which will create a concern for the antibiotics, the initial antibiotics that he had consumed, where the mechanism through which these superbugs are developed. One is intrinsic or natural, where the tolerance observed in the genus level and the species level, and by acquiring mutation or gene transfer, cross resistance, even through cross resistance, your super power antimicrobial would have become or would have reached the level of superbug. Cross resistance, co-resistance are one pattern of becoming superbug. So the AMR mechanism, which the entire biosystem or the entire ecosystem has been involved, where uh, every individual and every living organism, both biotic and abiotic, uh, has an hand in transferring the superbugs within the community. Is AMR abnormal? If the question is raised to me or to many of the scientific researchers, we say that it is natural and it is a rule and it is not an exception, because the same field we say that AMR is not abnormal, it is mere survival of the fittest that the organism tries to do. Diseases. So these are the list of well-known antibiotics which has been prescribed across the globe to certain infections that are caused by microbes. It's a 4,000-year old medicine vicious cycle where we say that in the era of 2000 BC, people used to, people say that along with the roots, though they do not say or they try to subplay the game. Along with the rules, they say that you say, I mean, they ask you to recite certain prayers so that it gets cured off. By 1800, people give you a drink or a potion, which a chemical based formulation, which will try to cure you. 1900, the same formulation has become a pill where they ask you to swallow and then the patient gets cured of. By 1950, the era started where antibiotics were ruling the world. By 2000 AD, antibiotics have been exhausted. Now we again are back into our usual form where we survived millions of years by depending only on the natural sources. So now people again say, eat the roots. Eat the roots indirectly, get certain products, natural products, and that could be used as a uh, slides, or the next few slides will try to decipher you or help to decipher you to identify and explore the natural products that is present in various floras and faunas. The very first thing is dereplication process, where we search for the natural products.
Guys, 48.
So the microbes, deptomycin from MRSA, and plants this particular dereplication process. One is the cost. The cost of doing such experimentation is specific diseases or disorder. So the result is going to be a very narrow spectrum. The target will be eventually caught, and the infection will surely be subsided if I'm going to have a 100% efficient natural product based antimicrobial is being developed through dereplication process. The second is search for a broad spectrum, where you have where we are looking for the entire spectrum of microorganism. The microbials have been developed against both gram positive and gram negative, or even a species level, and genus level identifications were being made, and the antimicrobials act against the entire spectrum of gram positive and gram negative bacteria. The only limitation that has been put forth would be the cost involved in exploring it and identifying the best natural product out of the spent amount. The advantages: the treatment can be prescribed at a very early stage because the antibiotics is going to work for the broader spectrum, and it will have the ability to treat multiple disease. However, though the initial investment is costlier, but it is cost effective because uh, the candidate may not run behind one single set of antibiotics when it works for a broader range. The third process is changing the culture conditions. By doing so, we ourselves can start developing a pattern of antimicrobials, where a simple alteration in the culture conditions will end up getting antibiotics or antimicrobial. Antibiotic era has not yet found out one new other antimicrobial since then. So REST all involves ribosomal engineering, high throughput elicitor screens, co-culture cultivation, and genomic mining. All these are the ones where or the ones which helps you in deciphering the natural product patterns. For each and everything, there is a mechanism or the pattern through which we can easily get the antimicrobials. In case of genetic engineering, we try to knock out the gene and start to see whether any antimicrobials have been produced. And co-culturing cultivation is very much similar to your the previous one. Here you grow one species along with the other, and there will be a development of resistance with one on the other. Last uh, two methods is high throughput elicitor screening and genomic mining, where we try to uh, obtain the novel compounds by understanding the metabolic pathways which have been involved in forming secondary metabolites. By that, we can easily figure out which could be and screening against non-growing persistent cells and screen for inhibitor and virulence. All these methods comes under the unconventional screening methods, which eventually helps in identifying the antimicrobials. So a scientific glance, brucellosis is a zoonotic disease caused by Brucella species, which is a gram-negative bacteria, and it has been a neglected disease in India. It is usually seen in cattle, but there are chances which gets jumped onto humans also. Specifically, when the organism is going to, when the organism such as Brucella abortus and Brucella mellitinus is there, it is easily seen in humans infecting or humans causing brucellosis. And this particular transfer usually happens when and different parts of the plants are previously been reported in both Ayurveda and Siddha for treatment of major diseases and ailments. And whereas the aqueous extract is able to give more or less near to 11 plus or minus zero point 0.2, which could say that the aqueous extract is performing well, and Brucella mellitinus is very much sensitive to your uh, Tarina asiaticus aqueous extract. So learning about the MIC at 30 mg per ml, we were able to see a positive clearance for Brucella mellitinus. So this is the uh, research work that we have done, and it has been published in South African Journal of Botany. So to conclude the entire topic and give an opening for the rest of the speakers for the next 2 days, I would just suggest that the investment on AMR activities and further researches and innovations are required to control this particular antimicrobial resistance. We need to provide awareness and understanding of AMR through effective communication, training, and education. We do need to strengthen the knowledge and provide evidence to surveillance. Reduce the incidence of infection is a way which can be achieved through effective infection prevention controls. Last is optimize the use of antimicrobial agents in health, animals, and food, which is the most important thing that needs to be taken back home by us, so that we reduce the your attendance sheet. Come feedback session is also been available in YouTube, which needs to be filled up for the certification process. Thank you. We are just looking for the questions. If anything is being posted on the query box, we'll just answer it down.
ஆமாம் என்கிட்ட இல்லையே அது இதே நான் அவனுக்கு காமிச்சு விட்றேன் ஒரே செகண்ட் ஒரே செகண்ட் ஒரே செகண்ட்